So see here, this cell wall is required so that it will provide a definite shape to the bacterial cell. If you remove the cell wall, the rigidity of the cell or the bacterial cell will be lost, and the protoplast, which is having a spherical shape, will come out. So how you will obtain this protoplast? You simply need to treat the any bacterial cell which is having cell wall or peptidoglycan with the penicillin drug. This penicillin will inhibit the cell wall synthesis. And if you incubate this particular cell and penicillin in this isotonic medium, ultimately you will get this cell which is sphere in shape. This is called protoplast. So protoplast is a plasma membrane which is surrounding your cytoplasm of the cell. So normally this is the proof that the cell wall is required, this rigidity is required so that the cell can maintain a definite shape and the size actually. Okay, so different shapes also we had discussed in yesterday's class. Mostly the bacteria are found in four general shapes. So one is coccus, one is rod shape, for example E. coli, and other is vibrio and spirillum, or the helical shapes. Okay, so these are the basic shapes. Beside this, other bacteria also occur in some other unusual shapes also, some exceptions. Okay, so so the topic of interest here, so we are going to discuss is the cell wall. The cell wall, how the cell wall is synthesized, how this is required for the structural maintenance or the rigidity. Okay, so first of all, the students who were present in yesterday's class know that we have discussed this particular topic where we have discussed the cell wall or the peptidoglycan is made up of two structural subunits, NAM and NAG, and they are connected by a glycosidic bond that is beta 1,4 glycosidic bond. So this is important now. There are two subunits. One is NAM and NAG. They make a disaccharide unit, and each time this disaccharide will be added either this side or this side of the chain when the chain will grow. Now the most important of all is not only this beta 1,4 glycosidic bond which is required for the structural maintenance or the cell wall maintenance. There are other cross links are also established between these amino acids. So generally there are 5 amino acids. I also told you the trick yesterday. So there is a subunit called NAM, N-acetylmuramic acid, to which 5 amino acids are linked via one lactic acid residue. So this is lactic acid. This is connected via this third carbon of this NAM sugar. This lactic acid then connecting 5 pentapeptide, which is made up of 5 amino acids. So the trick is L-alanine, D-glutamate, mesodiaminopyruvic acid, or in some cases you will see the lysine is present instead of mesodiaminopyruvic acid. Then there are 2 D-alanine at the last. And most importantly, this last D-alanine will be removed when this D-alanine make up link with other chain of peptidoglycan cell wall. So see here, this is one chain. So that's how the one chain will be formed. And in this way, many different chains will be synthesized for peptidoglycan cell wall. And each time you will see that the penta peptide side chain is attached to NAM residue. So this is, first one is alanine, this is glutamic acid, glutamate, third one is mesodiaminopyruvic acid or lysine, and last two are alanine again, D-alanine. So there was a trick to remember this. So we can write it like this: ICA A. And always remember this also, they are always found in this form, LDL GD. So this is L-alanine, D-glutamate, Lysine is actually L, or in some cases you will see the mesodiaminopiamelic acid is present. I will tell you that in what and which cases the lysine is found, in which cases the mesodiaminopiamelic acid is found. And last two are the D-alanine. As from the previous discussion, you know, mostly the sugars are present in D form in nature, whereas amino acids are found mostly in L form. But the bacterial cell wall is an exception where you will find that certain amino acids are also present in their D form. So this is the best example. Okay. Now, when different chains are synthesized inside, okay, over the cell, then ultimately in the last step the two chains will come. So this is one chain, so let me annotate this is one chain which is growing in both the directions. So this is one chain, and this is other chain, so this is second chain. Similarly, it is growing in both the directions. Now these two chains, and suppose there are hundreds of chains which make these cross links, or this is called transpeptidation step. This step is very important for the rigidity of the cell wall, so that the structural maintenance of the cell wall. So in gram negative bacteria, so first of all we are talking about gram negative bacteria. So in gram negative bacteria there are generally diaminopyamelic acid, and in gram positive bacteria you will see there are lysine instead of this diaminopyamelic acid. This is one difference. The second difference is this D-alanine, either this or this last D-alanine, which is the fourth one, and actually the fifth one is being removed when this cross-linking is taking place. So this D-alanine, which is at the fourth position, will make a cross-link with the third diaminopyamelic acid of the other chain. Similarly, in gram-positive bacteria, instead of direct linking, there is one pentaglycine interbridge, which is required for the cross-linking between this chain and some other chain of NAM and NAG residues. Okay, so this last, or the penultimate, this is called penultimate, that is fourth one actually. So fifth D-alanine is removed, then penultimate is before second last actually. So this will make a bond with L-lysine, which is present in gram-positive bacteria, and thus they will make the cross-linking. So this last step is very important for making a rigid cell wall in bacteria. Now, the thing actually I missed yesterday. Uh, okay, so this I want to tell you in the today's class also. Here we were discussing that there are NAM and NAG when we are talking in terms of peptidoglycan in bacteria. But in archaea, this NAM has been replaced by NAT, N-acetyl talosamine uronic acid. And there are certain differences in amino acid sequence also. So here it is in archaebacteria. So this is pseudomurin or pseudopeptidoglycan structure. So in archaea, it is found. So first of all, there is N-acetyl talosamine uronic acid. There is no NAM, and this is NAG. So this is very similar as it is found in the peptidoglycan cell wall. So this is exactly the same. Only difference is it is having this NAT instead of N-acetyl talosamine uronic acid instead of N-acetyl muramic acid. And the second difference, as you can see here, they are connected via beta-1,3 glycosidic linkages. So there is beta-1,3 glycosidic bonds. And this beta-1,3 glycosidic bond is very resistant to lysozyme. So lysozyme will not able to hydrolyze archaeal cell wall. Also, see here, when this cross-linking is being formed, so their amino acid sequence is quite different. Instead of alanine, they are having glutamate or glutamic acid first. There is no, you can see here, there is no lactic acid present between these amino acids and these sugars. So directly they are connected via glutamic acid or glutamate, then alanine, lysine, then again glutamate in some cases, and then alanine. So this is a sequence. There is a change in amino acid sequence, and when when this linking, cross-linking has been taking place. So this is one chain. This is growing in both the directions. And this is second chain of pseudo-peptidoglycan, which is also growing in both the directions. Uh, they will cross-link between a positively charged amino acid lysine and negatively charged amino acid glutamate. So that's how they are more stable cross-links between these two amino acids. Okay, so this is one, the structural rigidity of the cell wall is better. Second thing is they are having modified lipids. Therefore, this archaebacteria can survive in harsh environmental conditions in which most of the bacterial species cannot survive. Now the question is this okay, so this is our peptidoglycan cell wall. So the question is, this is always like DAP in gram negative and lysine in gram positive, and this cross-linking in gram negative is always direct cross-linking, and in gram positive it is always having a peptide interbridge. So this is not true in some. There are some exceptions also, but most of the cases they follow this trend. Okay, so there are some exceptions too. Uh, So there are variants of peptidoglycan are often the characteristic for particular group, and therefore they are having some taxonomic values. So they can be used to classify the microorganisms or bacteria depending upon these differences. Most gram-negative bacteria have amino acid composition that is cross-linking, shown in figure 3.13a, where this diaminopyramidalic acid is making a cross-link with the penultimate or the second last alanine. This variant is also observed in some gram-positive bacteria also, for example, Bacillus, Clostridium, Corynebacterium, and Mycobacterium. Okay, so also in nocardia. So these are some exceptions. Bacillus, Clostridium, Corynebacterium, Mycobacteria, though these are gram-positive, but they will have this particular cross-linking pattern where D-alanine will cross-link with the diaminopyramidalic acid, which is very similar to the gram-negative bacteria. So you need to remember, Mycobacteria is very common. So this is having trend of gram-negative bacteria and cross-linking. Other gram-positive bacteria substitute the diaminopyramidalic acid for lysine or mesodiaminopyramidalic acid in some cases. And these cross-links chains are via interpeptide bridges, which may be glycine pentapeptide bridge, for example. Okay. Instead, some examples are also given here in which there are certain other differences has been reported. So instead of lysine, they may have this type of pattern. So the cross-linking between these two, lysine and D-alanine, can be through either glycine pentapeptide bridge. This is glycine 5. If you're not able to see, I can zoom it further. Okay, so this is glycine 5 is shown here. This is one. In some cases, there is L-alanine and there are 4 to 5 glycine. Okay, so they may be used to connect this L-lysine and the alanine. In some cases, there is L-alanine 3 times repeated, and this tripeptide is used to bridge these two chains. In some cases, L-alanine, serine, threonine, and glycine, serine, alanine can be used to link the two peptidoglycan chains to make it rigid. Okay. And in some cases, as shown here, they can be replaced by ornithine. This is one of the amino acid which is also present in urea cycle. So the examples are given here also. So for first, this is seen in Staphylococcus aureus. So this is one here. So this glycine pentapeptide bridge is found in S. aureus. Second one is Staphylococcus epidermis, Micrococcus roseus, Streptococcus thermophilus, and so on. There are many different microorganisms, gram-positive bacteria, which show this type of pattern of cross-linking. Okay, so this is all about how the cell wall is different in gram positive and gram negative bacteria. Though they both belong to eubacteria, therefore a general peptidoglycan cell wall is found there. But in case of archaebacteria, this is quite different. They are having beta 1,3 glycosidic bonds. Also, their pattern of cross-linking is completely different. Therefore, you can put archaea in a completely different domain because they are also differ in their 16S rRNA signature sequences as well as their cell wall and lipid patterns. There is a query. Okay, so let me answer this. Oh, fine. Okay, so first now we will see how this peptidoglycan cell wall is being synthesized. Okay, so here. So this peptidoglycan is a polymer that contain the two so it is starting from here. So let me start it from here only, though I already told this particular paragraph many times in my class. So the bacterial cells almost always are bounded by chemically complex cell wall, which are required for the rigidity as well as for the cell structural maintenance. The cell wall protect the bacteria against osmotic lysis. So this is also required because generally the bacteria are found in aqueous environments. And if there is a aqueous environment, so the water can go inside the cell can be entrapped inside the cell, and the cell will lyse as a result of osmolysis, as you can see here also. So see this. So suppose the cell wall is absent, or you have treated so mostly the cell wall is absent in one group of bacteria that is called mycoplasma or PPLO, pleuropneumonia-like organism. They do not have a cell wall. Or somehow you make an bacteria which do not have a cell wall. If you treat bacteria with penicillin, ultimately the cell wall will not be synthesized and the protoplast will remain. This is also called L-form bacteria. So L-form bacteria is bacteria which is lacking. L for lack the cell wall. So this is protoplast. If you transfer this protoplast to an hypotonic solution, so hypotonic medium is something when the concentration outside is less than inside of the cell. So what will happen? The water will go inside the cell. It will go across the plasma membrane, and cell will swell due to H2O influx. This is called endoosmosis because the water is going across a cell wall, across a cell membrane, plasma membrane. Sorry. So ultimately what will happen, cell will swell, and when the osmotic pressure inside the cell is extensive, the plasma membrane is unable to hold the osmotic pressure, and ultimately it will lyse, so undergoing lysis. So you can, from this discussion, it is evident that this cell wall is required to protect the bacteria from osmotic lysis. Okay, again we will move to our pathfinder. Oh shit, I closed this. So wait a minute. Sorry, actually I closed the window somehow. Okay, so we were here. So this peptidoglycan is called murin, and in gram-positive bacteria, it is quite thick, 20 to 80 nanometer thick, homogeneous layer, which is present outside the plasma membrane. In contrast, gram-negative bacteria having a thin plasma, this peptidoglycan layer, this is only 2 to 7 nanometer thick, which is very thin actually. And this peptidoglycan cell wall is covered by an outer layer or outer membrane. This is 7 to 8 nanometer in diameter. Okay, covered by a 7 to 8 nanometer thick outer membrane. So gram-negative bacteria is actually having two membranes. One is normal plasma membrane and one is outer membrane, which is having some toxic component. We will discuss them in detail. Peptidoglycan is a polymer containing two sugar derivatives. One is this NAG and second is NAM, which is important. So NAM will come first when we are talking this in this way, beta 1,4 glycosidic bond. If you put NAG first, then you will say beta 4,1 glycosidic bond because it will be in reverse order. Okay, so keep in mind whether you are asked in this order. So if you are having NAM first, then this is beta 1,4. If you are having this NAG first, and it is beta 4,1. Okay, so keep this in mind. Joined through this beta 1,4 glycosidic bond, NAM is they are having lactic acid attached by an ether link. So this is ether, ROR. A peptide chain of four alternating D and L amino acid is called tetrapeptide, which is connected. But actually, these are five amino acids which are connected to this. This is talking, and actually, here he's talking about a mature peptidoglycan chain where the last D-alanine is removed, actually. Okay, it's just before cross-linking. So, what is the procedure? How this is synthesizing inside the bacterial cell? Let's discuss that because this we already have discussed. This step is called transpeptidation step. In many gram-positive bacteria, there is an acidic substance present which is called teichoic acid. So let's see what is this teichoic acid. This is also important. So it is given here. So this is teichoic acid. So you can so this is plasma membrane. This is thick peptidoglycan cell wall. So you can think like, because this is very thick, as you can see. So this is obviously gram-positive bacteria. Whereas in gram-negative bacteria, this is thin actually. And there should be an outer membrane. So here it is gram-negative. So from the thickness of this cell wall, you can tell whether this is gram-positive or gram-negative. So this is very thin in case of this is peptidoglycan cell wall, and this is thin in case of gram-negative bacteria. Also, gram-negative bacteria is also having this outer membrane. So there are distinguishing features between gram-positive and gram-negative. Let's discuss this teichoic acid. This teichoic acid is a polymeric substance. So you can see these rods; these are polymers. So this teichoic acid can either link to the peptidoglycan cell wall. Okay, or they are linked directly to the plasma membrane, as you can see here. So if they are connected to lipids of plasma membrane, then they are called lipoteichoic acids. It is written here, lipoteichoic acids. And if they are connected to the plasma membrane, sugars, so they are called teichoic acids, simply. So now here again, so this is a acidic substance which is negatively charged, teichoic acid, in their cell wall. Teichoic acids are polyol. What is polyol? So this is polyol. So this OL is actually for hydroxy group, which are having multiple OH groups. Phosphate polymer, so also having this phosphate. And due to this phosphate, actually this is having a negative charge. So this phosphate is present so why this is called polyol? It is having OH groups. So it is having OH group means either it is glycerol or ribitol. Okay, so these are also sugars. And also they are having attached phosphate groups. This is a polymer of glycerol or ribitol and attached phosphate groups. If you are not able to understand this, please look here. So this is a teichoic acid structure where these phosphate groups are interconnecting links between these glycerol or ribitol. So this black color structure is either glycerol or ribitol, whereas the phosphate groups are connecting these together. So this type of structural repeats are called, and this complete substance, complete polymer is known as teichoic acid, which is made up of phosphate, glycerol, and a side chain, which is either R or R dash. Okay, so this R. This R may be anything, either glucose, D-alanine, or maybe other molecules, depending upon type of microorganism or type of bacteria. So because this phosphate group is present, so therefore they are having this negative charge. Now you can think like what can be the advantage of this teichoic acid polymers which are present in gram-positive bacteria. So gram-positive envelope is having teichoic acid, and these teichoic acid are having negative charge. So they can bind to positively charged metal ions, for example, magnesium and sodium. So what will happen if they bind with magnesium and sodium? They will provide an extra stability and rigidity to the cell wall. So this is the function of teichoic acid, which is written in your pathfinder also. So bearing a strong negative charge because of these phosphate groups, they can covalently link to the, okay, so this is also important. So they are linked to the N-acetylmuramic acid, NAM, of peptidoglycan layer or to the lipids of the plasma membrane. Again, important. So you need to remember if they are linked to the plasma membrane lipids, not a problem. They are called lipoteichoic acids, whereas generally they are present, they are linked to the NAM residues of the cell wall, peptidoglycan cell wall, just like the amino acid uh, are linked to the NAM residue only. So here also we have seen that the NAM is linked to these amino acids. Okay. And the teichoic acid is also an acidic substance, so it is also linked to this N-acetylmuramic acid. Okay, so the combination of teichoic acid and lipids are actually referred to as lipoteichoic acid. We told this. Teichoic acid are absent from the gram-negative bacteria. They do not have teichoic acid. Main function of teichoic acid to provide the rigidity to the cell wall by attracting the cations or positively charged ions like magnesium and sodium. So this we have discussed. Again, the same structure which I showed you. Actually, we have seen the colored version, so we can skip this. Among bacteria, certain types of cells have no cell wall at all, and either they have very little cell wall material, so these are called mycoplasma or PPLO, pleuropneumonia-like organisms. There is one complete genus which generally lack a cell wall. Members of this genus of bacteria do not have cell wall, so mycoplasmas are therefore smallest non-bacteria which can grow and reproduce outside the living host cells. Now, the question is, what is L-form bacteria and what are the similarities between mycoplasma and L-form? So, mycoplasmas are naturally do not have a cell wall generally, whereas these L-form bacteria are synthesized, so they also known as L-phase bacteria because they are do not having a cell wall, they lack a cell wall. L4 lack. Cell wall deficient bacteria or they lack the cell wall. L form can be generated from many bacterial species which are usually having the cell wall. As I showed you, if you treat the bacteria with penicillin, they are not able to synthesize the cell wall material and the cross-linking will not take place between the chains of peptidoglycan. So ultimately, they do not have a cell wall, therefore they're called L-form bacteria. So for example, Bacillus subtilis E. coli, because they are E. coli is gram-negative, you should know, and Bacillus subtilis is gram-positive. So if you treat them with penicillin and you grow in an isotonic solution, they will not able to synthesize the peptidoglycan cell wall, and therefore they're called L-form bacteria. Now, you need to keep in mind there are two things. One is gram-positive bacteria, one is gram-negative bacteria. So if you see this particular diagram will help you understand this particular concept. So suppose this is the gram-positive bacteria and you treat this particular cell with penicillin. So this cell wall will not be synthesized and you will only have this plasma membrane and the cytoplasm inside this particular plasma membrane. So this is normal protoplasm, you can see. But suppose if you treat a gram-negative bacteria with this penicillin, what can happen? So suppose this is the structure of gram-negative bacterial envelope. What will happen if you treat the gram-negative cell with the penicillin? The cell wall will not be synthesized, but they will have this plasma membrane and this outer membrane. Okay, so because the two membranes will be in close proximity and therefore they are something called spheroplast, which is given in your book also. So, okay, so this is the difference between protoplast and spheroplast. See here, the plasma membrane and everything within the plasma membrane is called protoplast. Protoplasts lack a cell wall. If gram-positive bacteria are incubated with antibiotic penicillin in an isotonic solution, protoplast formation will occur. But penicillin inhibits the peptidoglycan synthesis. But if gram-negative cell retain their outer membrane after treating with penicillin, and therefore they are classified as spheroplast because some of their cell wall will remain inside this outer membrane. Okay, so there is a difference between spheroplast and protoplast. So protoplast are the are obtained from the gram-positive bacteria, whereas spheroplasts are obtained from the gram-negative bacteria if you treat them with antibiotic penicillin. Now, since I'm showing this particular diagram, there are certain important points to be noted here. First of all, you should know that this outer membrane is responsible for the toxicity of the gram-negative bacteria. And because this will not allow the certain drugs to enter inside the cell, okay, gram-negative cells. But most of the low molecular weight molecules can cross this outer membrane. It is more permeable in permeable structure when compared to this plasma membrane. As you can see here, there are certain porin proteins. These porin proteins are a kind of channel which will allow the low molecular weight molecules to cross across this outer membrane. So this is quite porous when we compare this outer membrane with plasma membrane. In plasma membrane, there are generally less porins, or they are absent from this particular plasma membrane. Therefore, they are less porous. Second thing is uh, they are having LPS, lipopolysaccharide, and these lipopolysaccharides are made up of three components. We will discuss this also in detail, but for now, you should remember this LPS is made up of three parts. This lipopolysaccharide is a large complex molecule contain both lipids and carbohydrates. So first of all, the composition is these are lipo carbohydrates. So these are glycolipids, glyco for carbohydrate, lipid for lipids. Okay, so consists of three parts: lipid A, core polysaccharide, and an O side chain. The most important part for toxicity is lipid A. So the LPS in Salmonella species has been studied the most. This lipid A region contains the two glucosamine sugar derivatives with three fatty acids and phosphate or pyrophosphate, which is attached to this lipid A. And so this LPS contributes to the negative charge on the bacterial surface because the core polysaccharide usually contain a charged sugars and phosphate. This lipopolysaccharide also helps to stabilize the outer membrane structure because lipid A is a major constituent of the exterior B flat of the outer membrane. And therefore, this LPS may contribute to the bacterial attachment to the surfaces and formation of biofilms. You know biofilms? So certain bacterial species can survive inside a plaque like structure. So this is called biofilm. So generally, more than one bacterial species are found in biofilms. A major function of the LPS is also to help create a permeability barrier. So it is a kind of permeability barrier. So it will not allow the antibiotics, bile salts, and other toxic substances that might kill the bacteria or will injure the bacteria, will not allow them to pass through the outer membrane. Okay, this lipopolysaccharide. And this O antigen is responsible for the eliciting the immune responses by an infected host. Now, the most important part is this lipid A portion in the LPS, which is toxic actually. So the toxicity of gram-negative strains is due to the presence of lipid A component in the LPS. LPS can also act as an endotoxin because causes some of the symptoms that arise in gram-negative bacterial infections. So, for example, septic shock, which is having no direct treatments. Okay, so this can happen. Whereas these porin proteins, which are present in the outer membrane, they are kind of channels through which the molecules which are having less molecular weight, less than about 600 daltons, can pass across the outer membrane. However, large molecules, for example, vitamin B12, also cross the outer membrane. But the mechanism is not known how the vitamin B, because it is having high molecular weight, but it is able to cross the outer membrane. Such large molecules do not pass through the porins. Instead, specific carrier transport molecules are also present in the outer membrane, which allow them to pass through the outer membrane. But for normal survival of gram-negative bacteria, this outer membrane is important because it will not allow the drugs to pass across the, and therefore this lipopolysaccharide, which is the red colored substance here, is a kind of permeability barrier in the outer membrane. Okay. Now there are certain lipids which are present, these proteins, which are called Brown's lipoproteins. So what are the Brown's lipoprotein? You can see they are connecting links between outer membrane and the peptidoglycan cell wall. They're called Brown's lipoprotein. They're also important. They're also responsible for the structural stability and maintenance of gram-negative envelope. Okay, Brown's lipoprotein. If someone asks you about Brown's lipoprotein, so they are mainly connecting the inner leaflet of the outer membrane with the peptidoglycan cell wall. So they're present in this pyroplasmic space. Okay, so responsible for structural maintenance. Okay, now we move back to the topic of discussion. So we were here actually. So we have started this. Then alpha bacteria. These alpha bacteria are generally protoplast or the spheroplast, which can be derived from the bacteria, which is generally having the cell wall. But if you treat them with penicillin, they will lose the cell wall, and ultimately they are called alpha bacteria. This is done by inhibiting the peptidoglycan synthesis with the antibiotics or treating the cell with the lysozyme. This is actually discussed in the enzyme class. So lysozyme is a kind of enzyme which is present in our secretions, tears, saliva, and so on. It is a specific protease. Sorry, this is a specific enzyme which will cleave the beta-1,4 glycosidic bond in your peptidoglycan cell wall between NAM and NAG residues. So the mechanism also we have discussed. The mechanism is through carbonium ion or an oxonium ion formation. It is requiring one aspartate and one glutamate residue for catalysis. So this mechanism we have discussed in our previous classes. You can find in your enzyme lecture. Okay. So an enzyme that digests the cell wall, peptidoglycan cell wall only. As L-form has no cell wall, its morphology is different from that of the strain bacteria that which is derived from which it is derived. Okay, so because this cell wall is important for the structural or the morphology of the bacteria, if you treat with the penicillin or some drug which is inhibiting the cell wall synthesis, the shape of bacteria will be completely different from the bacteria from which this L-form bacteria has been derived. Typical L-form cells are generally spheres or spheroids. Okay, so this also we have discussed from here. So if they lose the cell wall, they are generally spheroids, sphere-like shape normally, because they will try to maintain this cytoplasm inside the plasma membrane. Mycoplasma lack a cell wall, but these are not considered as L-form bacteria. So please note this, mycoplasma also do not have a cell wall, but we cannot call them L-form bacteria because L-form bacteria are derived from the bacteria which generally have a cell wall with the treatment of some drugs. And they are not derived from the bacteria that normally have a cell wall. Therefore, they are completely different genera. Now, biosynthesis of peptidoglycan, this is important topic. So we will cover in today's class only. So the cell wall, biosynthesis of peptidoglycan divided into following steps or stages. First of all, the donor molecules. So the donor molecules are either UDP, NAM or UDP, NAG. So you should need to remember this. First stage is the cytoplasm and lead to the synthesis of nucleotide sugar-linked precursors. So these nucleotide sugar-linked precursors are the donors which will donate their NAM and NAC components to make a proper cell wall. So these are donors, UDP-linked sugars. In the second step or stage, the sequential addition of the amino acids, which will always occur to this particular donor UDP NAM, occur as a result into the formation of NAM pentapeptide. So first of all, this pentapeptide will form, but during cross-linking, this will remain tetrapeptide because last D-alanine will be removed. So the third step is, which is taking place at the cytosolic membrane of the bacteria. So these two steps are actually occurring in the cytosol of the bacterial cell. So suppose this is a bacterial cell. Okay, so actually I need to change the screen. This is bacteria. This bacteria is having a plasma membrane. So this is plasma membrane and this is cytosol. The first two stages of cell wall synthesis are actually occurring in the cytosol. Then the third step is actually occurring in the inner leaflet of plasma membrane. And then only this, when the cross-linking is actually taking place, this cell wall components or the chains will be deposited here so that they can make the cell wall structure outside the plasma membrane. So you need to remember the position of their synthesis also, because sometimes they are asked as direct questions. Okay. So from here, oh sorry. So here, the third stage, which is actually taking place at the cytosolic membrane, cytoplasmic side of the plasma membrane, in which UDP NAM pentapeptide, which is being formed from the NAM UDP NAM, make a complex with the bactoprenol. This is bactoprenol is present in the plasma membrane of bacteria and forming a component that is called lipid 1. Okay, so see here, this is the plasma membrane lipid bilayer in bacteria, and this is cytosolic side. Okay, so in the cytoplasm, this UDP NAM will come and the 5 amino acids will be attached to make UDP NAM pentapeptide. Then UMP from this UDP, which is diphosphate, monophosphate will be released and one phosphate will remain attached to the NAM. Okay, so you can see it from here. This bactoprenol phosphate is generally present in the inner leaflet of bacterial plasma membrane, to which it will make a this is phosphate link. So phosphate to phosphate linkage. This bactoprenol phosphate will be linked to the phosphate and with the NAM and pentapeptide. This complete structure is called lipid 1 when this bactoprenol is linked to NAM pentapeptide. Then what will happen? UDP NAG, which is the donor of NAG in peptidoglycan cell wall, will come from the cytosol and it will be added to NAM. Let me annotate this. So this NAM is not having NAG up to here, up to the lipid 1 structure is formed. Then this will be converted into lipid 2 when this NAM will be linked to, okay, so this UDP NAG will donate this NAG and this UDP will be removed. Okay, so this will go and this NAG will come here and will be linked to NAM by one beta 1,4 glycosidic bond. So this bond between these is actually beta 1,4 glycosidic bond. So suppose you are treating some cell with penicillin. So this beta 1,4 glycosidic bond will be broken down as soon as it is formed. So therefore the cell wall synthesis will not take place and therefore they are called L form of the bacteria. Means the bacteria which do not have a cell wall. They can be of two common structures, either protoplast or spheroplast. Okay, so now you are able to understand what is lipid 1, what is lipid 2. So the only difference is If it is having NAG and acetyl glucosamine, then lipid 1 will be converted into lipid 2, and they will have a bond beta 1 for glycosidic linkage or bond between these two. Okay, so this lipid 2 then will be flipped towards the outer side of the plasma membrane. So this is plasma membrane here. If it is flipped outside, so it will be going like this, it will flip, so that this particular material, which are the chains of these peptidoglycan, will be deposited outside the plasma membrane, and they will in the final stage they will be cross-linked, so that us okay, so the cross-link I should make with some different colors. So these are chains of NAM and NAC. So each time these disaccharides will be added to these chains. Okay, so then there is cross-linking. So let me make this cross-link like this. So these cross-links are formed between these chains, so that a rigid cell wall can be synthesized out of these polymers. Okay, so this type of cross-linking is also required to make a proper structure, proper cell wall, rigid cell wall. So here we yield lipid 2. And then in the fourth stage, the peptidoglycan biosynthesis will take place at the outer side of the cytoplasmic membrane. It will be flipped towards the outside of the cytoplasmic membrane, involve the polymerization of newly synthesized disaccharide. So each time these disaccharides will be added to the inner side, and they will flip towards the outside, so that they can be added to make single chains of peptidoglycan cell wall. So it involves polymerization. But remember, each time when this is happening outside the cell, always a disaccharide unit, peptide unit will be added to the chain to make it growing. Okay, incorporation into the growing chain of the polypeptide. Finally, this last fifth step is called transpeptidation. So during transpeptidation, dealanine, these are the last bond is broken, and this last dealanine is removed. So this on one stem, peptide is first cleaved, and enzyme substrate intermediate is formed, which is concomitant release of the terminal dealanine. So the last or the terminal dealanine is lost from one chain. Okay, so here the cleavage reaction provides the energy. So now you can ask me a question, how this is happening, how this energy will be coming. So because this bond is broken, so dealanine, which are the last two amino acids in the pentapeptide, this bond will be broken, so that they can get the energy, essential energy, to make an X cross-link. So this bond is broken, and this dealanine, which is released, this will provide the energy which is necessary for the transpeptidation reaction to happen, which occur outside the cytoplasmic membrane. So this is happening outside the membrane. So because the cell wall is present here outside the cell membrane of the plasma membrane. So a second step involves the transfer of peptide moiety to an acceptor. This acceptor is a non-alpha amino acid group or a dibasic amino acid in the second stem. Generally, uh, you can have seen the diaminopyamilic acid is a kind of acceptor to this. Let me show you. So the reaction resulted in the formation of a new peptide bond between the penultimate dealanine, this is the second last dealanine, and a donor peptide, and an amino group in the cross bridge of an acceptor peptide. So this acceptor can be direct diaminopyamilic acid, or in some cases, if this is not going to accept this, this will have some crossing bridge, for example, glycine pentapeptide bridge, or some other common bridge which we have discussed in previous slide. Okay, so they can be used to cross to make the cross link, as we seen here. So either glycine pentapeptide bridge, or alanine glycine 4 to 5, alanine 3, and so on. These bridges can be used to link, cross link, or can be used in transpeptidation step. So fine. Okay. Wait a minute. If you have any queries, please ask me so that we can start the staining. Actually, after this, I want to. So, a question has been asked in previous year, actually. So the question is like this. Okay, so you can also ask me this. Generally, we are treating a bacterial cell with penicillin to inhibit the cell wall synthesis. Exactly how, what is the mechanism of action of penicillin? So the answer is the penicillin will inhibit DD transpeptidase. So this transpeptidase is required for transpeptidation step. So if the transpeptidase enzyme will be inhibited by penicillin, it will not able to cross-link these chains. So see here. So these crosslinks will not be formed. Okay, so either this or this. As a result, the bacterial cell wall will be susceptible to lysis because it will not be as much as rigid as it should be. So simply what will happen, the uptake of water will lead to osmotic lysis. More easily, the cell will die as a result of osmotic lysis. This is an indirect way of killing the bacterial cell. Okay. Second thing is, okay, so let me scroll. Okay, so fifth step is transpeptidation itself. In this particular step, Trishla is having a query actually. So see here. So the fifth step is actually the transpeptidation step. So what will happen actually, as we have seen, there are 5 amino acids which are linked to UDP NAM. So that the structure is formed is called pentapeptide. Okay, so this is also visible from here actually. So this is the complete structure actually. Okay, so the peptidoglycan chain. So this is a chain. Suppose this is running in both the directions, and wherever there is NAM, this is an acetylmuramic acid. So this yellow color sugar is linked to lactic acid and the 5 amino acids, the alanine, glutamic acid, mesodiaminopianoic acid, or lysine, and the 2D-alanine at the last. So what will happen when the cross-linking will happen? So the last D-alanine, this particular D-alanine will be lost. This will be removed. Now this removal means removal need this particular peptide bond will be broken, And this particular peptide bond, when broken, will provide the necessary energy which is required for the transpeptidation step. So what is transpeptidation step? Now see here in the next page, actually it is shown clearly. So suppose there was five amino acids which were linked to NAM, and the chain is grown over the outer leaflet of plasma membrane in bacteria. So the chains will be like this. So this is one chain. Suppose there is a second other chain and so on. There are many chains which are growing. Now suppose this is one chain. Let me zoom it out so that you see. Now this is one chain and this is second chain. So each time this disaccharide unit will be repeated. So this is occurring in both the directions. Suppose it is running this way, this is running this way. But each time this disaccharide unit will be coming. So suppose there is disaccharide, then again there is disaccharide. So this is growing in this way in both the sides, okay? And so on, sorry. Fine. And so on. Similarly, there is some other chain here which is also growing in both the directions, and which is a long chain. But whenever there is a NAM, suppose here is one unit called NAM and NAG, and here is also NAM. So each time this NAM is attached to this peptide, which is actually 5 amino acid long. But when this cross linking has been taking place, so during this formation of this particular bond, the last D-alanine is removed, and the energy which is released from the breakage of peptide bond is used to make this particular second bond, so that these two chains can be linked to each other. What is the benefit to linking these two chains? You can think like many ropes, okay, many threads. But to make a rope, you need to wrap them around each other so that they can be more structurally rigid than a single thread. So each thread is actually very fragile. It will not be so much stable, but these cross-linking will provide extra rigidity to the complete cell wall structure so that it can be maintained under aqueous conditions, or in so that it will avoid any osmolysis in bacterial cells. Okay. So this is written here. So in gram-negative bacteria, mostly this D-alanine, this is penultimate D-alanine or the second last D-alanine, which is present at fourth position, will be linked to diaminopyruvic acid. Whereas in gram-positive bacteria, the linking is through a bridge or some peptide again. So this is S aureus. So in S aureus, so glycine pentapeptide bridge is used to link this D-alanine with the lysine. Now see here. So during transpeptidation, this D-alanine, which is actually the last D-alanine residues which is added. So see here, this is 1, 2, 3, and 4 stages. So again, if this is not clear, so it is clear from here also. So you can think in one step, this will coming, second step, this is coming, and at the last step, when these 5 amino acids are being added to NAM residue, the D-alanine will come together. Okay, they are not like they will come separately, they will be coming jointly here. So this is shown here in the first diagram also. So they are coming together. So to make the pentapeptide, okay. But this last D-alanine should be removed when there is this transpeptidation step is happening. Okay, so this D-alanine bond in the stem peptide will be cleaved first, and an enzyme substrate intermediate is formed. So the enzyme which is some peptidase which is responsible for removal this particular D-alanine, it will break the peptide bond and will make an enzyme substrate intermediate complex with the concomitant release of this terminal D-alanine. So this D-alanine will be removed, but it will be bound to this penultimate D-alanine so that it can be cross-linked to some other chain. cleavage reaction provides the necessary energy for the transpeptidation reaction, which occur outside the cytoplasmic membrane. So this is the plasma membrane. This reaction will happen on this side of the plasma membrane because this is cytoplasmic side and this is extra cellular. Okay, so this is happening here. Occurs outside the cytoplasmic membrane. A second step involves the transfer of peptidyl moiety to an acceptor. So in this case, the acceptor is either diaminopyruvic acid, see here, this mesodiaminic acid of the second chain. So suppose this is the second chain. So here this is diaminopyruvic acid, which is the acceptor. This will be the donor. So enzyme will remain bound to this D-alanine, and last D-alanine is lost. So it will link this particular D-alanine to this diaminopyruvic acid because this diaminopyruvic acid is acting as an acceptor because it will have an extra NH2. So from this it is more visible. So this is mesodiaminopyruvic acid, this B structure. So this B structure you can see here, this is alpha carbon, this is having COH, NH2. This is also having one alpha carbon here, which is having NH2 here also. So this side group, R group, and the mesodiaminopyruvic acid will act as a receptor. So because it will have a lone pair of electrons, so will accept this particular alanine. So this particular bond will be formed between the two chains, and this is kind of indirect linkage between this chain A and this chain B in the peptidoglycan cell wall. Okay. But suppose if diaminopyruvic acid is not present, it is absent, which is generally happening in gram-positive bacteria. They have L-lysine instead of diaminopyruvic acid. Then what will happen? This will not have this extra acceptor arm. So what will happen here? Peptide interbase or some other interbase is used to link this alanine. This is second last alanine of one chain with the third L-lysine of the second chain of NAM and NAG residues. Okay. If you are not able to understand it still, I will explain it again, but after the class only. Okay, so let me finish the second topic also so that we can move on. So here we have discussed the monomorphic and pleomorphic organism yesterday. Most of you were absent in the yesterday's class, so I do not know who were present in today's class. So Anchal, yes, she was present. Yogita, Yashraj. Okay, so Yashraj and Trishla were absent in the yesterday's class. So actually, we have discussed about bacterial cell. There are certain structures. So the bacterial cell is having a plasma membrane, a DNA containing region in the center that's called nucleoid, is having a capsule, or this capsule is actually the cell wall. Uh, Actually, sometimes the capsule is a substance or the polymer which is accumulated over the cell wall. Okay. So, and this is very important, mesosomes. What is mesosome? Mesosome is an structure of the plasma membrane which will increase the surface area of plasma membrane. So what are the functions of mesosome? First of all, it will increase the plasma membrane surface area. Second, it will sometimes regarded as mitochondria. So it will make necessary ATP. So the proton pumps are present in this particular structure so that it can be used to make necessary energy for the bacterial cell. Therefore, can be compared with the eukaryotic mitochondria. So we have mitochondria, but bacteria have these mesosome-like structures. Also, this mesosome has been reported in taking part in DNA replication and site of many enzymatic reactions, some specialized enzymatic reactions. So you can note it is very important and sometimes has been asked directly. So what is the function of mesosome? So it is regarded as mitochondria of prokaryotic cells. It is having some role in DNA replication. Oh, sorry. Replication and having some specialized enzymes. So enzymatic reactions. You don't ask me what are these enzymatic reactions. I really do not know what are these. Okay, so these are the some important applications and functions of mesosome. Okay, so you need to remember. And there are ribosomes. Ribosomes here are 70S. Okay, so which are made up of two subunits. One is called as 30S subunit, which is smaller subunit. Oh, sorry, 30S. And the second is actually 50S. So, oh, sorry, I do not know why I'm writing like this. So these are made up of two subunits. Now, generally, the question is asked in the terms of sRNA, sorry, this rRNA. So it is having only one rRNA. So 16S rRNA, whereas the 50S subunit is having two different rRNA, 23S rRNA, and actually it is also having 5S rRNA. Okay, so this is all about ribosome. This you need to remember. Okay, so one more question has been asked from this particular discussion. So in bacteria, the ribosome is responsible for translation. Okay, because these are the machines where protein synthesis has been taking place in bacteria. So this 23S is actually important in transpeptidation reaction. So you can think like when the new coming tRNA with then charged amino acid will come, so it will be added with the help of this 23S subunit. Okay, so you need to keep in mind this. Then there are flagella, cilia, and these like things, flagella, pilus, and they are appendages of the bacteria which are having different functions. Mostly this flagella is required for the locomotion. As I told you, they are having two kind of locomotion, either run and tumble. So what is run, what is tumble? Uh, so there is a query, is linkage of D-alanine with DAP-R group. Okay, wait a minute. So actually it is, so since this mesodiaminopyruvic acid is having this particular structure, okay, so and because these two are engaged in formation of peptide bond with the first and because this mesodiaminic acid is present between alanine and glutamate. Okay, so they were engaged in formation of peptide bonds, obviously. So this is with this NS2, yes. So what we were discussing, actually, I forgot. So staining. We were discussing the structure of bacterial cell. Okay, so we were here. So this flagella is having one kind of motion that is called run and tumble, in which it will run and then it will change its direction so that it can change its direction, and it will move in some other random direction. So this is kind of motion has been seen in which is made up of a long run and then change in direction. It is tumble. When this flagella move anti-clockwise, so generally the bacteria run, and if it move clockwise, so mostly bacteria will change its direction. So you need to remember this. So this by flagella is mostly, so this is a motor flagella, and it will move like this. Whereas the flagella in eukaryotes is not having the same motion, it will have some whiplash-like motion. So it will move left, right like this. But in flagella and the bacteria, it will having a rotatory motion. Okay. Now there are some other structures in bacteria also. For example, pilus. Pilus is required for the DNA transfer. For example, so here it is given actually. They're having fimbriae, pilus. So these are different structures. Pilus is elongated hollow appendage used to transfer the DNA to the other cell. They are called sex pilus. Whereas fimbriae are other hair-like structures are present over the bacteria. They are required for adhesion to the surfaces. So suppose bacteria want to adhere to some surface in our respiratory tract. So it requires these fimbriae-like structures to hair-like structures, appendages to stick to the host substrate or surface. So they're different, they are having different structures, they are having different functions. So you need to remember this. And flagella, they are moving the cell forward. And other structures are also given, and their functions are also described here. So glycocalyx, slime layer, capsule, uh, inclusion bodies. We will discuss inclusion bodies in detail in our next class. So please do not worry. They are stored nutrients. They are used for bacteria for so and for just like you have glycogen in your body. So they are energy stores in liver and muscles. Bacteria have these inclusion bodies so that they store their nutrients in these inclusion bodies. Okay. So they are phosphates or other substances like polyhydroxybutyrate, so that they can be stored inside the cell and can be used whenever required. Rest we have discussed. Mesosome, actually plasma membrane fold into the cytosol and increases the surface area. Can be regarded like mitochondria of eukaryotes and also involved in the DNA replication and so on. And this also we have discussed the gram-positive and gram-negative structural differences. It is having a thick peptidoglycan cell wall, which is generally 20 to 80 nanometer thick. Peptidoglycan is also called murin. And these are called lipotecoic acid and teichoic acid-like structures which are present over the cell membrane of gram-positive bacteria. They are having some negative charge, so they will bind some positive charge cations so that provide the structural stability and rigidity to the cell wall. Whereas teichoic acid is absent from gram-negative bacteria. Gram-negative bacteria is having an outer membrane, and outer membrane is having some, these yellow colored substances here are lipopolysaccharides, LPS. So these lipopolysaccharides are having some O antigens also, which are required for immune reactions and so on. So our body is able to recognize the gram-negative bacteria only depending upon these O antigens. And they are toxic. They secrete some endotoxins. For example, lipid A component of LPS is actually toxic. So that can create problem in humans. The cell wall is quite thin, only 2 to 7 nanometer thick in gram-negative bacteria. And they are also having an inner membrane. But see, this is more porous in outer membrane is having some porins, so it is more porous. Generally, molecules which are having molecular weight less than 600 Dalton will be passed across the outer membrane. Now there are certain differences between gram-positive and gram-negative bacteria, which is described here also. Gram-positive bacteria, 90% of the cell wall, and it is generally thick, 20 to 80 nanometer thick, and they are linked by peptide bridges, generally 5-glycine residues as we have seen. Whereas in the case of gram-negative bacteria, they are either directly linked D-alanine with diacyl glycerol or diaminopyramidalic acid with the 2 tetrapeptides. Okay, 2 tetrapeptides because they are a last peptide in the pentapeptide is removed. D-alanine is removed in this transpeptidation step, and this is thin, 2 to 7 nanometer thick. Teichoic acid, it is present only in gram-positive bacteria. The gram-negative bacteria do not have the teichoic acid because we know teichoic acid is a negatively charged polymer. 10% of the cell wall is polyribitol, polyglycerol, phosphate. So teichoic acid, what is the chemical composition? It is polyribitol or polyglycerol phosphate. This phosphate will provide the negative charge to the teichoic acid. Lipoteichoic acid, if the teichoic acid is linked to the lipids of the plasma membrane. Periplasmic space is smaller in case of gram-positive bacteria, but periplasmic space, because there are two membranes in gram-negative bacteria, so they are having some enzymes for transport, degradation, and synthesis. Outer membrane, their gram-positive bacteria do not have an outer membrane, but they have, and they are having lipopolysaccharides also. Lipopolysaccharides, yes, the lipid A is endotoxin, core polysaccharide, and O antigen. This LPS is made up of three components in gram-negative bacteria. Now, depending upon flagella, you can classify the bacterial cells in, this is called monotrichous. This is lophotrichous if a bunch of flagella is present over one pole of cell. If they're present over both the poles of cell, they are called amphitrichous. And if the flagella is present all over the body of this bacteria or this bacterial cell, these are called peritrichous. So depending upon flagellar positions, they can be classified. Depending upon shapes also, they're coccus, bacillus, curved, either this vibrio or spirillum, or they are pleomorphic. Pleomorphic means when the bacteria is found in more than one forms. Also, there are differences between spirillum and spirochete, because the spirillum is a rigid body and spirochete are more flexible and they're quite elongated. Okay. We will discuss spirochetes because they are responsible for some of the infections, common infections. We will discuss in our upcoming classes. Then different bacteria and their shapes are given. Most important were the streptococcus. Strepto means the chain of bacteria is present. If the chain of bacilli is present, they are streptobacilli. Okay, then this is 1D. In one direction only, the bacterial chain can elongate, either this way. If they are moving in two directions, then they are called tetrads. They are arranged like four, like squares of four bacteria over the corners of a square. If they are present over the corners of a cube, then this type of arrangement is actually called as uh, sarcinae. So you can think of a cube here, and they are present over the corners of the cube. So this kind of model is actually called sarcinae. Sarcina ventriculi is an example of this. Whereas actually this is 3D. So if it is multiple planes, so they make a cluster, and this is very similar to the bunch of grapes. This is called staphylococcus. Staphylo means they are present in bunch. So strepto chain, staphylo bunch, sarcinae cube, and tetrad is normal for bacteria present over columns. So these are some basic differences between you need to know while giving nomenclature to the bacteria. And we have discussed these in yesterday's class. We also discussed everything. Okay, so because two were absent from the yesterday class, we discussed there are two big bacteria found in nature. One is Thymargyrata nambiensis, which is the largest. And earlier it was thought that Epulopiscum fischelsoni was the largest, but actually this is the second largest bacteria over Earth. Okay, so this Epulopiscum fischelsoni is having some cucumber-like shape, whereas the Thymargyrata nambiensis is having a round shape. Okay, so this is Thymargyrata, and this is Epulopiscum. Epulopiscum is also reported to give birth to living offspring. So this is showing some viviparity. This is also an exception in bacteria, because bacteria generally divide by binary fission. So the next important thing is staining. What is staining? Staining is a biochemical technique which can be used to color the specimens. Uh, Different type of dyes has been used. Some of them are cationic dye, some of them are anionic dye. So the simple question has been asked in one of the previous year papers. So you need to remember from here the two type of dyes: basic dyes and acidic dyes. Okay, so they are given here actually. So the basic dyes are crystal violet. So this is being used in gram staining method. Methylene blue, saffronin is counter stain and gram stain, and malachite green. All are basic dyes. So the question was whether crystal violet is an acidic dye, basic dye, neutral dye, or none of above or all the above kind of question. So if you have studied that crystal violet is a basic dye, you need to remember this in this format only. So the crystal violet, not only crystal violet, methylene blue, saffron and malachite green, all are basic dyes, whereas acidic dyes are eosin and acid fusion. You can remember acid fusion because this is acid here, but if the fusion is only coming like this, fusion, so the eosin and fusion are acidic dyes. Now, there are different type of methods, three basic types. One is called simple, differential, and specialized methods. Simple method, if you stain a specimen, so all the cells are uniformly stained. There is no difference. Simple stains react uniformly with all the cell types which are present and only distinguish the organism from the background or the surrounding. Uh, Differential dyes or stains do not stain all the cells with the same color, but they are able to discriminate different cell types. For example, gram positive and gram negative, and gram stain is actually a differential stain. Not only gram stain, the acid fast stain is also a differential stain. So because we are able to differentiate acid fast bacteria from non-acid fast bacteria. Specialized stains are able to detect the specific structures of the cells. For example, we used to stain the flagella and endospores. Okay, so most important, flagella, we can stain, and endospores, we are able to stain with the specialized dyes. So if we want to stain some specific structure of the bacterial cell, then we require specialized stains or dyes. Okay, so here, so if you want to stain the endospores, so okay, so if it is visible, so we need a dye that is called Skaffer-Fulton dye. Is used to endospore the first stain with malachite green in the presence of heat, and then with saffronin. So a combination of malachite green and saffronin is used to stain the endospores, and this is called Skaffer-Fulton method or procedure, or the stain can be called a Skaffer-Fulton stain, which is made up of malachite green and saffronin. So this can be a direct question, which stain can be used to stain the endospores, and what are the components which is being used in these particular stains. Second is, if we want to stain the capsule, so it can be stained with India ink or nigrosin dye, which is generally used to stain the polysaccharides which surround many bacteria and some of the fungi. Okay, so the capsule stain with the India ink or nigrosin dye, it is a kind of negative staining. And if you want to stain the flagella, flagella is staining is taxonomic value, because if there is a flagella, and as we have discussed many different positions of flagella, monotrichous, amphitrichous, and peritrichous, and so on, or lophotrichous. So they should be stained so that we can count number of flagella and exactly where they're present. So the bacterial and archaeal flagella are fine, but they okay, so they're seen under electron microscope, because in light microscope you cannot differentiate the flagella actually uh, very very clearly. So although bundles of flagella can be visualized by dark field microscopy, you can see bundles, but actually to separately see them we need electron microscope. So what is the, so here we need a method that is called gray method. And in this method, we stain this with, uh, okay, there are two methods actually. One is Lefosen method and one is gray method. So fusion is a dye which is used to stain the flagella, or it is stained with para rosanilin and this Lapson method. Okay, so these two methods are common, but generally basic fusion dye is generally So fusion is actually two types: basic fusion and acid fusion. Okay. So if fusion come in some question, it should be mentioned that whether it is acid fusion or basic fusion. Generally, basic fusion is used in Gray method, and it is very common to stain the flagella. As we have read from the text, so this fusion is written here acid fusion. So this is an acidic dye, but if it is written like basic, so basic fusion is used to stain the flagella. So this should be remembered. Now I hope we are running out of time, so we will discuss gram staining in upcoming class. So that's all for today. If you have any question or query, you please ask.